Welcome to today's Explainer. Now, I want you to really treat this session as a masterclass, because today we're diving into the absolute foundations of biological science, specifically the Unit 3 introduction and beyond. We are going to journey all the way from the broadest, most philosophical mysteries of the living world right down to the fundamental microscopic building block that makes every single living thing possible, the cell. You see, biology, at its core, is the study of living organisms. But to truly understand life, we have to look way past the overwhelming, chaotic variety of plants, animals, and microbes we see every day, and instead seek out the unifying principles underneath it all.
So here is our roadmap for today's Explainer. We'll start by unpacking the mystery of life. Next, we'll explore the incredible legacy of G.N. Ramachandran. Then, we'll break down a concept known as reductionist biology. After that, we'll clearly define the living world, which leads us straight into topic 8.1, our ultimate question for today: what exactly is a cell? All right, let's jump right in.
Section 1. The Mystery of Life.
For centuries, biologists basically just observed and described the form and appearance of living things. But think about it. From a towering redwood tree to a microscopic bacterium, the sheer variety of life on Earth is just incomprehensible. So as students of biology, we really have to ask ourselves this fundamental question: what underlying principle unites the immense diversity of living organisms? Seriously, if a mushroom, a blue whale, and you and I are all considered alive, what is the common denominator tying us all together?
Well, the answer is cell theory. It's cell theory that brings a sense of unity to this massive diversity, proving that all life forms actually share a cellular organization. But interestingly enough, this realization created a whole new mystery. You see, the physiological and behavioral processes of life, the actual things that make an organism living, require a profound integrity of cellular organization. If you literally break a cell apart, those individual molecules, they aren't alive on their own. The real mystery is how this intact, perfectly organized cell gives rise to the living phenomena we observe. And to understand how scientists started unraveling that, we need to look at the pioneers of structural biology.
That brings us to Section 2, G.N. Ramachandran's amazing legacy.
Let's trace his academic journey because it is truly remarkable. Born in 1922 in a small town near Cochin, India, his father was a local college math professor, and that early influence gave Ramachandran a profound, rock-solid grasp of mathematics. That analytical foundation was absolutely crucial. It propelled him to graduate at the very top of his Bachelor of Science honors physics class at the University of Madras in 1942. He then earned his PhD from Cambridge University in 1949, and just a few years later, in 1954, he published a groundbreaking discovery in the prestigious journal Nature, detailing the triple helical structure of collagen.
Because of this, he became widely recognized as a founder of the Madras School of Conformational Analysis of Biopolymers. I know, that sounds like a mouthful, but it was a literal game changer. By applying rigorous mathematical and physical principles to biological molecules, his school of thought completely, fundamentally transformed how we understand protein structures. So, the key here is how his physics background collided with the biological world. While at Cambridge, Ramachandran actually met the legendary Linus Pauling. Pauling's models of the alpha helix and beta sheet structures in proteins deeply inspired Ramachandran, pushing him to solve the complex structure of collagen, which, by the way, is the most abundant protein in our bodies. Not only did he crack its unique triple helical structure, but he also invented the famous Ramachandran plot. This is this brilliant, ingenious mathematical graph used to analyze the allowed shapes or conformations that proteins can possibly take. To this day, it remains one of the most outstanding contributions in the entire history of structural biology.
All right, moving on to Section 3, Understanding Reductionist Biology.
Now, Ramachandran's work is actually the perfect example of a broader scientific philosophy that really took off in the 20th century. Scientists realized that to figure out how the intact cell produces the mysteries of life, they had to, temporarily at least, take it apart. We call this reductionist biology. It's defined as a physico-chemical approach to studying and understanding living organisms. In simple terms, we take the strict objective rules and techniques of physics and chemistry and apply them to the messy, complicated world of biology. We reduce biological phenomena right down to their raw chemical and physical components just to make sense of them.
And this is executed very systematically.
Step 1, you take living tissues and analyze them using cell-free systems. Basically, you break the cell open to isolate its individual parts. Step 2, you analyze these parts to find out exactly what specific organic compounds are in there. And finally, step 3, the most crucial part, you ask, what are these compounds actually doing? How do these mere inanimate chemicals carry out gross physiological processes like digestion, defense, or even something as complex as memory? By answering that, we find the exact molecular basis of physiological processes, which, by the way, is also exactly how we understand and treat abnormal processes during diseases.
Okay, on to Section 4, defining the living world. So while reductionist biology is incredibly powerful for zooming in on the molecular machinery, we occasionally have to zoom back out to answer the most fundamental question in biology. When you look around, you see both living and non-living things. You've got to wonder, what actually makes an organism living? Where is the absolute defining boundary? What is it that a completely inanimate rock or a drop of water does not have that a living tree, an animal, or a bacterium does possess? The definitive answer is wonderfully simple. It's the presence of the basic unit of life, the cell. This is the absolute, non-negotiable prerequisite for life in all living organisms. If it has cells, it's biological. If it doesn't, it is inanimate. Period. Now, within this biological world, everything is composed of cells, but it scales differently. Unicellular organisms are composed of just a single, solitary cell that handles absolutely everything on its own. On the flip side, multicellular organisms like humans, plants, and your pet dog are composed of many, many cells working together in these massive, beautifully coordinated networks. Which brings us right into Section 5. This is Topic 8.1, the absolute core of our masterclass. What is a cell? To truly define a cell, we usually look straight at unicellular organisms because they prove a vital, undeniable concept. A single cell is fully capable of two things. Number 1, independent existence, and number 2, performing the essential functions of life, things like respiration, metabolism, and reproduction. And here, my friends, is the golden, unbreakable rule of biology. Anything less than a complete structure of a cell does not ensure independent living. You can have all the right DNA, the right proteins, and the right lipids sitting in a test tube, but if they are not organized into a complete, intact cellular structure, independent life simply cannot happen. So, let's lock in our formal definition right here. The cell is the fundamental structural and functional unit of all living organisms. It's structural because it is the literal physical building block, and it's functional because it's the smallest enclosed environment where the chemistry of life successfully and independently operates. But getting to this level of understanding took centuries of technological evolution. It kicked off way back in the 1600s when Antony von Leeuwenhoek first saw and described a live cell using some very early rudimentary lenses. Fast forward to the 1800s, and scientists are starting to peek inside the cell, with Robert Brown famously discovering the nucleus. But it was the continuous improvement of microscopes, culminating in the invention of the electron microscope in the 1950s, that finally blew the doors open. That technology revealed all the intricate, mind-boggling structural details of the cell membrane, the organelles, and the internal matrix that actually make life possible. As we wrap up today's explainer, I really want to leave you with a profound thought to chew on. We've learned today that reductionist biology can break a cell down into entirely non-living chemical components. Yet, the cell theory strictly states that a complete cellular structure is the absolute baseline for independent life. So I want you to ask yourself, as we assemble proteins, lipids, and DNA molecule by molecule, at what exact miraculous moment do we actually cross that boundary? When does a complex arrangement of inanimate chemicals suddenly become a living, breathing, independent entity? Think about that as you continue your studies. Thank you so much for joining this masterclass.